Well, hi everyone. As you can tell, I have not gotten better, and my voice is coming in and out. So just bear with me as we go through this little presentation. Hopefully by Thursday, it'll all be back to normal. So we're already in week 9. We're in the last 2 weeks of new material for this course. So let's take a look at what we have going on this week. So we're going to have get into week 9 material, as well as have our final regular lecture exam. So first off, let's talk about the lecture exam. So the lecture exam will take place on Thursday, 10 a.m. to 11 a.m., just like normal. It covers the lecture material from week 7 and week 8. So that's covering chapters 9 and 10, mitosis and meiosis, as well as chapters 11 and 12, which is the Mendelian genetics and chromosomal basis of inheritance. So it's a 4-chapter. It's quite a bit of information. Just like last time, it's mostly multiple choice with short answer questions. Moving into week 9. So in week 9, we're going to take a closer look at DNA replication. So we learned in in chapter 9, the cell cycle, where we have the G1 phase and then the S phase and then the G2 phase before we actually go through mitosis or meiosis. So the S phase is when the DNA actually duplicates or replicates itself. So chapter 13 focuses in on how that occurs. So what exactly that process is for replicating our DNA. We'll also go into a little bit more detail about how mutations occur within our DNA and how that can end up affecting the protein that is being made, which affects the overall trait. So as always, good idea to do the overview. Make sure you read through the objectives and understand them before you take the quiz this week. We also have chapter 13 material. So for Campbell book users, it's chapter 13. For OpenStax users, it's chapter 14. With our study guide, lots of terminology as normal. And then we also have some, good visuals and some good questions. I'd like you to pay close attention to this question at the very end. So here you're going to diagram the DNA as it's replicating and be able to draw out and identify all of these different structures. This will help you out a lot in understanding what each one of these roles is in the DNA replication process. So what is the leading versus the lagging strand? What's the replication fork? What are the Okazaki fragments? What do they do? What's helicase versus topoisomerase and so forth? So diagramming it out will help you visualize everything. And then we have some test yourself questions. Take a close look at the videos as well to give you some good visual aids of the processes. I have 3 different videos for you for my instructor videos that follow chapter 13 of your textbook. We have our good supporting videos as well. And then our practice problems as well. Okay. We won't have a chance to work on chapter 13 in our recitation since we'll be taking an exam. So just going over some of the information. We have multiple choice for the first part. Okay. Be careful. Some of these questions are select all that apply. So notice the circles instead of the A, B, C, D, E. So there might be more than one answer. So you'll have to have all answers correct in order to get full credit. We'll also be looking at how we can clone DNA in order to uh, either treat genetic disorders or maybe make more insulin for a person to be in, that has diabetes or things like that. So you're going to learn about that, the gene amplification process with the PCR reaction. So you'll write that down. And then we have a matching section. So here are those terms again, like the helicase, the leading strand, topoisomerase. So you'll see, match the term with the information here. Not all choices will be used. So just be aware there's like 10 choices, but there's only 8 questions. We have some YouTube video that you'll watch, answer that, and complete the information. So this is due just like normal Sunday midnight, and then you'll have your quiz Sunday midnight. Okay. For lab this week, we are doing what I like to call Punnett Palooza. So we're going to be focusing in on Punnett squares and how to set them up, how to properly do them. So it's a great opportunity for us to practice if we didn't quite get it from chapter 11 and 12. So our lab is, it's going to be pretty much just walking our way through all of the different parts of the Punnett square and setting them up for all sorts of different scenarios. We will also be looking at pedigrees, so setting those up as well and trying to figure out, what each of the components mean. So we'll spend a lot of time on that during lab this week. The exam itself, even though it happens before this lab, there will be some Punnett squares, but not too many. So I want you to, try to get as much of it down as possible before the exam, but just realize that you will also have this whole practice period after the exam to get you ready for the final exam. And then one other thing with the lab this week. Due Sunday midnight this week is our signature assignment that I've been talking about. So if you are going to print it out and hand it in person, it's due at lab. Otherwise, the digital version is due by Sunday midnight, June 11th. In addition, we have the skills test going on this week for lab. So half of you will go do the skills test with me. So I'll pull aside each person individually and test you on all of these different parts. Okay, so make sure you understand how to use the microscope properly and be able to measure things and all of that. So this will occur this week in lab. So half of you will go. I'll ask for volunteers first. If there's no volunteers or only a few volunteers, then the remaining spots will be randomly placed with students. Whoever doesn't go this week will go in week 10 or final week. If you have any questions in the meantime, feel free to email me. Otherwise, hopefully I will be more understandable on Thursday when we meet in person for our exam. So good luck.